Okay.
Lovely. Right. Hello, everyone. Thank you for joining us for the second live session of this year and this month, which is based on environment. So as you would already know from our social media posts, we are using one of the topics that you have selected, which is environment, as our themed month in January. And we have had a live session last week, and today's live session is a live talk. And today we are going to learn more about what Dr. Holly Shiels is doing at the University of Manchester. So Holly is a reader at the University of Manchester, and her research aims to understand how climate change actually affects fish physiology. So for those who are not very familiar with our platform and the structure of the talks, so we will first start with a talk from our guest, from Dr. Shiels, and after she gives her talk about her research, we will have a 10-minute comfort break and resume back to get answers to your questions. So during the break, please make sure you submit your questions to our social media accounts or by using the chat function on the YouTube channel, which should be on the right-hand side. So as I mentioned, Holly works for University of Manchester. So Holly, thanks very much for joining us today. It's an absolute pleasure to have you here with us. And thank you very much for sparing your time.
It's my pleasure to be here. Thanks for inviting me.
So would you like to start by sharing your screen and see if we're okay with
Sure.
everything?
Let's see if we can get this going. Hold on. Share screen. Okay, how's that? Can everyone see it?
Yeah, we can see it.
Okay, brilliant. Okay, well, thank you very much for inviting me to give this live with scientists lecture, and especially it's a privilege to be talking in the environmentally themed month. Obviously, the environment is very much on everybody's minds at the time, so hopefully this project or this presentation will be of interest. So I'm going to be talking to you about fish physiology, or how fish work in an era of climate change. And I was taking this image here. This is this is an image from the Fisheries Society of the British Isles. And a number of years ago, they hosted a conference specifically dedicated to how fish globally were changing or being affected by climatic differences. And I think it's a really stark image of the implications of the climate. And this is changes in temperature on the surface of the planet and how it affects the fish living in them. So I guess climate change is part of our reality, and we know it's driven by exploitation of land, sea resources, and reliance on fossil fuels. And we're expecting a minimum of about 2 degrees rise in temperature before the end of the century. We've got higher CO2 in the atmosphere, which is altering the pH of the world's oceans and making them more acidic. We've got eutrophication of coastal waters, so nutrient runoff in coastal waters leads to hypoxia, and hypoxia is low oxygen levels. And also our search for fossil fuels comes with the risk of oil spills, toxicants entering the atmosphere and the aquatic ecosystem in the form of pollution. And we can kind of think of these combined climate change stressors visually in this figure here. Well, here we have our burning of fossil fuels. It's creating the greenhouse gases that's raising atmospheric temperatures. That's causing higher temperatures in the oceans as well. And there's a direct relationship between temperature and oxygen saturation. The higher the temperature, the less water can hold oxygen. So directly, the effects of temperature are lowering the oxygen contents of our bodies of water on the planet. In addition, the exploration of fossil fuels causes pollution both in the atmosphere and in the aquatic ecosystem. In addition, agricultural runoff, so intensive farming and urban waste and urban industry, cause a thing called eutrophication. So this is when we nutrient load aquatic ecosystems, especially coastal ecosystems, and then microscopic organisms use up all that oxygen and all that nutrients, and therefore causing even greater reduction in oxygen saturation. And together then, these high temperatures and increased eutrophication causes this lower oxygen saturation, which we term hypoxia, or hypo meaning low, oxia meaning oxygen, so low amounts of oxygen. And so these are some of the combined stressors that organisms are facing in the environment. And in my lab in Manchester, we're really interested in how animals cope with these climate changes. How does an organism cope with changes in temperature, hypoxia, pollution, pH? And in particular, we focus a lot on aquatic organisms, and that's actually what my talk is going to focus primarily on today. So how do animals living in the aquatic environment deal with these different climate change stressors? And our lab is actually particularly interested in the cardiovascular system and how the cardiovascular system in itself is central to how animals cope with the environment. And we kind of consider the heart to be, I guess, the linchpin for all activities that an animal has to do. And certainly in terms of performance, that's true, because you need a strong cardiovascular system, the system that's pumping blood around the body, the system that's taking oxygen out of the environment and distributing it to the working and growing organism. And so performance is really tightly regulated to the heart. And so if we want to understand how the environment is affecting performance of organisms, then looking at the cardiovascular system or other aspects that are associated with the cardiovascular system is a pretty safe idea. And so some of the aspects of performance that we're interested in are things like metabolism. So that's the delivery of oxygen, the removal of fuels, and the consumption of energy in the growing organisms. Growth, we're interested in how animals and how climate change affects animal locomotion, swimming, movement, interactions like behavioral interactions, like reproductive activities. Because all of these performance indicators, which are arguably underlined by cardiac performance, can impact lifetime fitness. And here's an example of salmon jumping up in a stream. And so the temperature of that water, the cleanliness of that water, the amount of oxygen in that water, and the acidity of that water are all going to impact the ability of the animals to make this lifetime fitness jump upstream so they can reproduce and create the next generation. And so understanding how performance indicators and the cardiovascular system is impacted by climate change is one of the things we're really trying to understand in our lab. Now, before I go on, I wanted to say fish have a heart, but the heart is slightly different to the hearts of humans. So I wanted just to explain the difference. So here, the heart is made of 4 chambers in the fish. The first is the sinus venosus, where blood from the body is collected. Then there's a little pacemaker here that causes the heart to contract. That sends blood into the atrium. The atrium then sends blood down into the ventricle. The ventricle contracts and sends blood to the gills where it gets its oxygen. And I've got a little video here. The next one, this is a CT scanned image of a rainbow trout heart. And what you can see is these different chambers. So here's the powerful ventricle. You can see some of the muscular areas here. Here's the atria. And here's the sinus venosus. So this is where blood would come into the heart. This is now just taking a section through the heart so you can see the interior of the fish heart. And what you can see is the trabecular nature of the muscle walls. And this is where blood will be pumped out of the heart through here to the gills to get oxygenated. So I'm going to tell you 3 short stories about fish cardiac function in an era of climate change that have been going on in my lab. I'm not going to tell you all of the details, but I'm going to give you references at the end of the talk if anybody would like to learn more. First one is going to be about the brown trout, which is an endemic species in UK freshwaters, rivers and streams. Then we'll talk about a coastal species in the embryonic life stage. And at the end, I'll talk about a big ocean pelagic predator, the bluefin tuna. And we'll talk about stressors such as temperature, hypoxia, and oxygen or pollutants, and how they affect metabolism, swimming, growth, and morphology. So let's start with the first one, which is about the brown trout. So freshwater populations, especially in North America, changes in the population structure, changes in population performance have been linked to changes in climate change or changes in river conditions. There's also been mechanistic links between individual fish performance and climate change impacts on the heart. And so to bring this kind of riverine experiment to England and to Manchester, we're moving to, rather than thinking about big rivers in North America, we're thinking about this river catchment, which is the Ribble River catchment, which is in the area just beyond Greater Manchester, located here in the UK. And the work we did here is together with the Ribble River Trust, which is a local trust, people who are interested in, it's a charity interested in preserving the Ribble River and preserving the ecosystem for both the animals and humans who use it. And again, this study is going to focus on the brown trout, the Salmontrata, which is a salmonid similar to the species that you can see here in the migrations from North America. And in this study in particular, we were interested in looking at how temperature changes in this catchment, changes in dissolved oxygen in this catchment, and changes in water flow in this catchment were affecting the cardiorespiratory physiology and the movements of the species, the brown trout. So how do you go about doing this? Well, the first thing we had to do is get out into the river catchment and see what the climatic conditions were actually going on there. So this is a student of mine from a number of years ago. This is Jessica Lee, working with Gareth Jones from the Ribble River Trust. And they're in here doing electrofishing for brown trout, looking at different species that are in the river. They're measuring oxygen content, water flow, and temperature. What you can see, and this is the first figure I'm showing you, this is a figure showing, this is A and B are two different tributaries of the Ribble. And this is showing data from a temperature loggers we placed in the rivers. And you can see there's a slight difference between whether the temperature logger was placed in the shade or the exposed. And that's partly because this was part of a project the Ribble River Trust, it's hard to say that, was doing to look at the effect of riparian growth along the riverbank and how that would affect temperature and encourage trout back into the rivers. But what you can see is the winter temperatures are around 6 degrees and the summer temperatures are peaking just above 20. So information like this and information on water flow and oxygen dissolved levels that we get from the actual environment, we bring into the lab and then use that environmental data to set up our experimental studies in the University of Manchester. So in this next figure, what we're actually seeing is this is a fish in a treadmill. So this is a brown trout that's swimming. It has another fin. It's just tucked underneath. Here's the nose. You can see the slow body motion. This video is slowed down so that we're able to put markers on the fish and measure the kinematics of body motion as it's swimming. So what we've actually got here is a swim tunnel. So what you're looking down is top down to the swim tunnel of this treadmill for fish. And what we have is we put the fish in this working area. And we have a video camera recording above it. And then this is a environmentally regulated swim flume, so that we can change the oxygen levels in the water that the fish is experiencing. So we can create hypoxia. We can change the temperature in the water that the fish is experiencing, so we can simulate warming. And by changing the motor, we can change the speed at which the animal has to swim, so we can simulate velocity changes that occur with storms or violent weathers in different catchments. And so we looked at all of these to try and understand the effects of multiple stressors, these multiple climatic stressors, on the physiology of the brown trout. So here I'm going to show you a little bit of data. So this data slide is looking at one of our variables, which is metabolism. So metabolism is actually oxygen consumption. You can think of it as the amount of energy the heart has to pump around to deliver oxygen around the organism. And what you can see, so this axis is increase in metabolism or increased oxygen demand of the fish. And this axis is increased temperature. And you can see two different colors here. The orange is normoxia, so when the animals are being maintained at a regular oxygen, so 100% oxygen saturation. And then the blue is hypoxia when oxygen saturation is lowered to 70%. And the first thing that should jump out at you is that as you increase temperature from 10 degrees up to 20 degrees, we get an increase in metabolism. So metabolic rate increases regardless of any other condition. So that means that the heart has to spend more energy pumping blood around the body to deliver oxygen to supply that increased metabolic demand. The next thing I want to draw your attention to is the impact of water velocity. Now, these upper bars here are animals swimming at high speed, and these lower ones are animals swimming at a slower speed. And what you can clearly see then is that swimming faster also increases metabolism. So your heart has to work harder to swim at higher frequencies. And then if we look at hypoxia, which is the difference between these two colors at either the low or the high swimming speed or any of the temperatures, you can see very little effect. And in fact, there was no impact on metabolism for lowering oxygen in the water until we got to the most extreme condition of the highest temperature and the highest swimming speed. Now that's looking at metabolism. We looked at another measure, which was the kinematics, or the movement of the fish under these same conditions. And that data is summarized here, and it shows exactly the same pattern. That really, the real impact, this is showing the cost of locomotion. So how much does it take for the animal to move? And of course, movement is very important for energy expenditure, but also appropriate movement is important for migration, for predator-prey avoidance, for courtship rituals, etc. So understanding metabolism and cost of locomotion gives us quite an interesting picture of what's happening to these animals. And so the take-home message of this first little story I wanted to tell you about was that the brown trout in the river catchment seemed to be able to cope quite well with increases in temperature and with hypoxia and increases in water velocity at current levels. So the levels that we chose were the levels we were measuring in the water and in the environment at the time. We didn't push them to future levels. We wanted to understand what was happening right now and whether these were really stressing out the organisms. But it looks like metabolism and swimming were okay under these conditions, except when we got to the most extreme combined condition of high temperature and high water velocity. Then the lower oxygen started to have an impact on metabolism and on transportation. And so this shows that though the animals are maintaining right now, conditions are on the edge, because when you get to the extreme conditions of what's happening now, there are problems for these organisms. And so it shows that really action and policy must be put in place to prevent further changes in these environmental variables and to prevent additional stressors coming in. And on that point, one of the things we didn't look at was the impact of toxicants. But we know that though these rivers are in better condition, we still do have toxicant loads in the conditions in the river. And again, the increased number of climatic stressors that organisms face, the more difficult and more challenges their metabolism and their physiology has to overcome them. And before I want to move on, I want to point out one other thing. And that's that the oxygen saturation we sat in this study is at 70%. Now that is not a very strong oxygen challenge for fish, especially for brown trout. It's actually a percentage you would see in quite slow moving waters in warm temperatures in the river. So that's not a very strong hypoxic stress in this study. But hypoxia can get much, much worse than that, especially in the marine coastal ecosystem. So this figure here shows aquatic hypoxia, which again is low oxygen, or even anoxia, which is zero oxygen. And it shows how much of a pervasive problem it is in coastal environments. In a sense, you can imagine it as environments where there's no oxygen, so suffocation spots around the coast. And you can see these littered around the coast of places on the globe, especially where there is human populations, because again, it's the intense agriculture, urban waste, and fossil fuel use that's causing eutrophication and therefore hypoxia in these coastal environments. And of course, it combines with warmer temperatures and shallower seas in the coastal environments to again have these multiple stressor effects on the organisms living in these coastal zones. So that kind of sets the stage for the second study I wanted to talk to you about, and that is the embryonic cat sharks. And in this study, we're going to be looking not at adult stages like we did in the brown trout, but looking at what happens in these early life stages of the embryos. And again, we want to look at the combined stressors of temperature and hypoxia, and looking at them on morphology, growth, and survival. So the first part of the study was just to look at the effects of climate change on growth and development of these animals. Here is a lesser spotted cat shark. This is it inside of its mermaid's purse. You might have seen these washed up on the seashores. Here you can see one who's almost ready to hatch. There's the yolk. Well, no, it's about halfway through hatching, pardon me. Here's the yolk, which is feeding the growing organism. And here it is growing inside of its egg case. After probably another couple of months, it will have consumed all of this yolk, and the animal will actually break free and swim free. Now, the reason why these are pretty interesting animals to study the effects of climate change in the coastal environment is because these tendrils attach the animals to rocks or to seaweed in that coastal area. And that means that the animal cannot leave that coastal area. So it is sessile during development. And because these coastal areas do have higher temperatures and lower oxygen levels because of eutrophication in coastal waters, therefore these animals are actually experiencing and unable to leave these negative conditions. We wanted to know how these temperatures affected the growth and survival of the embryonic stage. But we're also interested in something else, and that is this concept of developmental programming. Because there's the idea that long-term changes in gene expression and phenotypic expression can occur in response to environmental conditions that occur during development. And so the idea was that maybe these animals living in these conditions during their developmental stage will program their physiology to give them either positive or negative impacts on or ability to cope with additional stressors in later life. And so we wanted to use the cat shark as a model because it's endemic to the UK. You can see them, so it's quite easy to measure growth and development, and they're a novel organism to look for. And the main things we're asking in this study is, does thermal, so temperature, and does oxidative stress, hypoxia, affect growth and development, and does it program cardiorespiratory physiology in later life? So in this one, our study design was quite simple. We have took shark embryos and we put them into one of 4 different environmental conditions. The first one was normoxic conditions, so 100% oxygen saturation at 15 degrees Celsius. We then had the same temperature, but lowered the oxygen to 50% this time, so lower than we did in the trout study. And then we had the same oxygen conditions, but we increased the temperature to 20 degrees. So we had 4 different conditions, and under this, we could look at the effects of climate change on the embryo's growth and survival. Once the animals hatched, we then put all the animals from all these different treatment groups into a common garden environment. And this common garden environment was at normoxic 15. And that's to simulate the fact that once the animals hatched and free swimming, it would be able to leave these potentially more hostile environments and search out an environment that would be ideal for its growth and survival. And we allowed these animals to grow until they were juveniles at 5 months old, and we culled them and studied their cardiorespiratory physiology to look to see whether or not any of the developmental programming or any conditions that were set during development were still visible after 5 months living in the same environment. Now, this part of the study is only partially done, so I'm just showing you some preliminary data. I'll focus on this part of the study as we go. So, what happened in terms of the direct effects of climate on the growth and survival of these animals? Well, the first thing is that the sharks grew faster, hatched earlier, but were a smaller size when they were reared at warmer temperatures. That can be seen on this figure. Here's body length on this axis and developmental time on this axis. And what you can see if you just look at these curves, the 2 warmer temperatures, the normoxia and the hypoxia in yellow and in orange. And you can see here's the growth of the animals from a very small embryo up to hatch. At this point, the animals held at 20 degrees hatched. So you can see this is maximum growth, hatching at about 20 weeks. The animals that were reared at 15 degrees, whether it be under normoxic conditions or hypoxic conditions, are shown in these colors here. And you can see it took these animals longer to actually develop. But the actual body size of these animals at hatch was greater than the body size of these animals at hatch. This is K, that's actually a growth curve. So it's actually telling you the actual growth constant of these growth trajectories. So the take-home message then is that if you incubate or if you grow animals, or animals are living in warmer conditions, they're going to grow faster and they're going to hatch sooner, but they are going to be smaller. And we found that quite interesting. I'll talk a little bit more about the implications in a second, but we also found it quite interesting that temperature had a much greater effect on these parameters than hypoxia. Temperature overwhelmed the impact of a 50% reduction in oxygen. Now, these animals are actually quite hypoxic tolerant, so we did know that this was not a huge stress on them, but this data doesn't really give you the whole story, because this is showing you the growth curves of the animals that survived. This shows you the actual survivability of the embryos under these different rearing conditions as a percentage of total number. And what you can see is that under the normoxic 15 group, all animals survived. At the warmer temperatures, a few animals died in the earlier stages, but under both of the hypoxic treatments, whether it be during either of the two temperatures, about 40% of the animals actually did not survive. And this suggests that hypoxia really was a challenge during development, but those animals that survived the hypoxic challenge were able to grow, depending on temperature, similar to their normoxic counterparts. And that's very interesting, and we're trying to understand what the difference between the animals that survived and the animals that didn't survive is. But again, regardless, it's really important to mention that size at hatch is a really important measure of life fitness. It interacts with the predator-prey interactions. The larger you are, the less somebody's planning on coming and predating on you or picking on you. And of course, as the animals grow, it can have implications on reproduction and other aspects of life history. So size is an important measure of fitness. And it's interesting to think that in this study, developing at warmer temperatures made you smaller. But hypoxia, though, did not impact the actual size of the animals that survived. So that's the first part of the study. Then there are direct climate changes on the embryonic growth and survival of the lesser spotted cat shark. But what about this developing programming? Is there any indication that these rearing conditions had an effect on animals after 5 months of being in the same condition? And again, this is very, very preliminary. We are doing further studies looking at gene expression in these animals. I'm just going to tell you a little bit about the morphological work that we've done. So the first thing to show you is this is a cross-section of the head of a shark. So here is a baby. This is a 5-month-old shark cut right through the center. So of course it's been euthanized. It's been embedded in formalin and then embedded in wax and then sectioned with a myotome. And this is a stain called H&E, and the muscle is in pink, and the darker colors show you nuclei in the muscle cells. And what we're interested in here was actually understanding the effects on the gills. So here is a gill arch. This is the first gill arch taken out of one of these sharks. And you can see here the gill lamellae, the primary and the secondary lamellae. And we measured a number of different parameters on the gills of the animals at 5 months old who've been kept for 5 months in the same conditions, but have been reared under different conditions. And one of the interesting things we were looking at was this thing called interlamellar cell mass. And this is the amount of this is cells that actually can gather in the gill lamellae. Now, if you think about it, the gill lamellae is where oxygen diffusion occurs. So the more open the lamellae is, the easier it is for oxygen to be transferred from the water across the gill and into the blood. This interlamellar cell mass shortens the amount of space on the lamellae that can be used for gas exchange. So we were interested to see whether or not there's any lasting effects of those different rearing conditions on this parameter. And here's some data here. And what this is really showing you is that the animals that were raised under hypoxia at 15 degrees, they had a significantly larger interlamellar cell mass to lamellar area in relation to their body size. And this suggests that they were being programmed for hypoxia tolerance. The next thing I wanted to show you was some cardiac morphology that we looked at across these animals. Now, this data is still very much in progress. We haven't even finished our data sets, but this is where we are to date. But what you can imagine is here's the heart of the sharks, and we took a section through the heart at this level here, and then you can section the hearts and look. So this is now looking, imagine this is the cross-section through the heart. This is the lumen of the heart. Fishes have a spongy myocardium, so they don't have a very big lumen. The blood kind of flows in and around these different trabecular muscle areas. And you can see there's a darker area on the outside of the heart, which is really important for contraction. And there's this spongy tissue inside. And what we found was that there was a change in the relative proportion of this spongy compact area in the animals that were reared at warm temperatures. So that's really preliminary days on the developmental programming, but I just wanted to give you some take-home messages from this study. Certainly, we know that warmer temperatures is causing sharks to develop faster, but be smaller at hatch. And we know this is going to have implications, or certainly could have implications, on fitness. Here's an example of this is the smaller at hatch, or this is the larger animal. No, this is the smaller animal because you can see the scale bars here. So this is the animal at 20 degrees. These are both hypoxic animals at 20 degrees, and this is at 15 degrees, both of them at hatch. Hypoxia didn't have that much of an effect on growth, but it did affect survivability. We have some preliminary evidence of developmental programming, which may be the first evidence of developmental programming in elasmobranchs, but we have a lot of work to do to really understand how these rearing environments and these different climatic conditions might impact later life fitness of sharks. Okay, so now on to the final story that I wanted to tell you a little bit about today, and that was moving away from temperature and hypoxia to the other climatic factor that I think is really important in the atmosphere or in the world today, and that's our use of fossil fuels and its impact on the ecosystem. So I'm going to tell you about a pelagic predator, the bluefin tuna, and the impact of oil on their cardiovascular system. So first of all, what's so great about tuna? So I've been working with tuna for years. I did work with them during my PhD back in Canada, out on ships in Hawaii, off the coast of California. They're absolutely amazing organisms. They're apex predators in the oceans. So they're very important for ecosystems. They have a unique swimming mode. They have regional endothermy, so they're able to keep parts of their body warm, which is fascinating from a physiology perspective. And they have a specialized cardiac physiology that underlies all of this. And these are some of the reasons why I just think tuna are fascinating animals to study. But that's not why we're studying them for this particular study. Before I go into the actual details, I wanted to make you think about how challenging it is to actually study the effects or the influence of the environment on an oceanic predator. How do you know where the animal is in its environment? Now, that used to be very difficult. People used to actually have to set fishing rods down, and they would know the depth of their rod, and they would know what animals they were catching at certain depths, and they could put probes at those depths, and they would try and correlate depth temperature, depth pressure, depth oxygen with where animals were being kept, animals being caught, to try and get an understanding of how these pelagic animals or animals that live in the deep or the open sea, how they were using and challenged by their environment. But that changed with the advent of biotelemetry. Now this is Barbara Bloch. She's a colleague in Stanford, and this is one of her programs, Tag a Giant. Here's her putting a telemetry tag in a giant bluefin tuna. Now, these tags give a lot of information. They tell they communicate with satellites saying where the fish are, so sending GPS, but they can also tell you the temperature, the depth, and some of the tags actually that are internal can tell you aspects of internal physiology, like muscle temperature and even acceleration, so some kinematics. So the reason why I'm telling you that is because it's important for this study about oil and these animals, because Tag Giant was involved in some tagging off the coast of America. So just to orchestrate you, here's Florida, this is the Gulf of Mexico, here's Newfoundland, and here's the coast of off coast of Maine in America. And this is where some tagging studies were done. So the white bars are where the animals were caught. Tags were put on these animals, and then they were tracked as they moved through the coast of America. And what's important is that these tracks tell us where they are at certain times. And you can see that this blue track shows that this particular animal was in the Gulf of Mexico at this particular time. This happened to be 2010, and this was April 2010. And we know that something else occurred in the Gulf of Mexico in April 2010, and that was the Deepwater Horizon oil spill. So if you recall, it ruptured on the 20th of April. It was capped nearly 90 days later, releasing about 5 million barrels of crude oil directly into the Gulf of Mexico. It's the largest accidental oil spill in the world. And what was quite unique about it was that rather than being an oil spill where you have a lot of oil on the surface, like when you have it from a tanker, a lot of the oil was horizontal in plumes and remained underwater. And therefore, it was directly interacting with the pelagic animals that were living in this pelagic deep water environment. And so we were interested in understanding how these apex predators were able to cope with this aquatic pollution, this oil spill. And so our study aim was to really understand why crude oil is toxic to fish hearts at the mechanistic level. So unlike the previous two studies where I talked about the first one in terms of swimming mechanics and locomotion, and the second one we were talking about growth and development, in this final little snippet, what I want to do is get to the crux of the matter, actually looking at the heart itself and the mechanisms of toxicity on heart function. So this was a collaboration between my lab in Manchester, NOAA, which is the National Oceanic and Atmospheric Administration, which is part of the U.S. Department of Commerce, and Stanford University. And here is the surface of the water a couple of days after the oil spill in the Gulf of Mexico. Now I'm going to take you on a tour from the whole fish down to what's happening at the heart cells of the fish. Here's our giant tuna. This is what the heart of the tuna looks like. And if you remember, I told you there's the atria, the ventricle, and the bulbus. So blood flow comes from the body into the atria, through the ventricle, and out to the gill. And what we do is we remove the hearts from the animals, and we perfuse the isolated heart with digestive enzymes. So these are things that break apart the collagen and everything that holds the heart together to liberate individual cardiomyocytes. And this is a confocal image of a cardiomyocyte from a bluefin tuna. They're very long, about 100 microns in length. And they're very thin, about 5 microns in length. And once we have these individual cardiac myocytes, we can start to really look at the cellular mechanisms that control function and how they could be impacted by toxic pollutants in the environment. This down here shows you the same cell, but now it's loaded with a calcium imaging dye that allows us to measure calcium inside the cell. And this is a calcium transient. Now we're interested in measuring calcium inside cells because calcium is actually the ion that's involved in the strength of contraction. Calcium has to come into the cell from outside to actually bind with the myocyte machinery, the contractile apparatus, to actually cause the muscle to contract. And so if you want to understand the strength and the rate of cardiac contraction, you need to understand the inflow and the outflow of calcium in the cardiomyocytes, because it's the inflow and outflow of calcium in an individual cardiomyocyte that causes that myocyte to contract and relax. And of course, it's the contraction and relaxation of hundreds of thousands of myocytes that causes the whole heart to contract and relax. And of course, then it's the contraction and relaxation of the heart that propels blood around the body and allows oxygen to be distributed to all the working muscles to allow movement, growth, and metabolism. So if you go down from the whole animal down to the very, very, very small area, calcium is a really key thing to study. We can study this with a technique called electrophysiology. And that's where we put a very, very fine glass needle onto the surface of the cardiomyocyte. The tip of the needle is about 1 micron thick. And we apply that there, and then we use it to measure the flow of single ions across the cell membrane. So we measure the calcium coming in, and that is directly proportional to the strength of contraction. Here's actually an example of the of a myocyte on a patch pipette. So this is a single myocyte isolated from the tuna. You can see it kind of moving here, and you can see that the each time the pipette, the electrical stimulus is given from the pipette, the myocyte contracts, and that contraction is calcium coming in across the cell membrane. The other thing we're interested in this study was to look at the combined effects of the action potential, so AP stands for action potential, and another ion flux, which is potassium. So if calcium controls the strength and the rate of contraction, it's the potassium and the action potential that control how the electrical impulse travels across the cell. And that's important because you know that there's a pacemaker in the heart, and that pacemaker sends out an electrical impulse. That electrical impulse is the action potential, and that action potential is what's required to allow that calcium to come in. And so these are intricately connected, the excitability of the heart and the calcium coming in. And so this study is to try and look at this level of cardiac function. And just to give you a sense of how it all works together, here in black is the action potential. So this is the activity of all ion channels opening in the cell membrane. That causes calcium to rise. So again, that causes calcium to rise inside of the cell. Each action potential is followed by a rise in calcium. That rise in calcium is followed by a contraction, which you can see here. And so this study was to examine these features and how they were affected by oil. Now, which oil to look at? So how do we, oil is a complex mixture of different chemicals.
Holly, and I'm sorry, I don't know if you can hear me.
Yes.
So this is quite interesting, and I'm really enjoying this, but I'm conscious of the time.
Yeah.
It's 6:45. I just wanted to let you know.
Oh, okay. Gosh.
Sorry. It's 7:15, not 2:45. It's 7:15.
Okay, no problem. I'll be. We're actually not too far.
All right,
okay, sorry about that. I hadn't looked at the time. Thank you.
Sorry for interrupting.
No, that's no problem at all. So specifically, work by my colleagues suggested that phenanthrene was the main component in oil that we should be looking at, and we looked across a number of different pelagic species, but I'm only going to tell you about the work in the bluefin tuna. So this first piece of data just shows you this is calcium. This is a measurement of calcium coming in. So the amplitude of this contraction is calcium influx. And what you can see is under controlled conditions, we have a certain amount of calcium, but when we add this component from oil, it reduces calcium influx into the heart. And that means that we've got less calcium influx, that's going to be less contractility, and that's going to directly reduce the contraction of the heart. The next thing we looked at was that electrical signal, the action potential. So again, here in black, we can see the controlled trace. But when we add phenanthrene, you can see that we're disrupting that electrical signal that's causing the action of the contraction inside of the heart. And you can see this here in the mean data, that the action potential duration is altered. That's pro-arrhythmic, it can cause arrhythmia. So the calcium can cause a reduction in force, and this electrical activity disruption can cause a reduction or an increase in arrhythmias. The last piece of data I was going to show you is this here, which is the potassium flux. And what you can see is here is potassium flux under control conditions. And again, when you apply this drug, it reduces potassium efflux. And that again is going to be causing a arrhythmia. And this is just showing the reduction in that potassium flux as you increase the concentration of this key component of oil. So take-home message number 3 is that fish that are exposed to crude oil or to polyaromatic hydrocarbons, which are in crude oil, causes a loss of contractile function. The heart stops contracting, and that's because of inhibition of that calcium current and that calcium flux at the single cells. We also know that animals exposed to these oil components have arrhythmic hearts. We know arrhythmias are associated with fatal heart attacks in humans, the same thing in fishes or related in fishes. And we know now that it's due to prolongation of the active potential duration and changes in potassium channel function that are caused by the effects of these polyaromatic hydrocarbons. And so certainly there is an implication of toxicants and our use of fossil fuels on animals in their environment and direct effects on the contractile function of these animals' hearts. And it's not just fish that we should be worried about, because in relation to our toxicants, these polyaromatic hydrocarbons, phenanthrene, they're globally distributed in both the oceans, but also in the atmosphere. And they're a huge proportion of what we're breathing in air pollution. And we believe that the similar effects that we've seen on the negative contractile effects and the increased arrhythmogenic potential in the fish heart is probably what's causing cardiac dysfunction in our hearts in poor air conditions. So then just to summarize, I've told you 3 short stories and I've given you only partial details to each, but I wanted to cover a breadth of the work we're doing to try and look at fish cardiac function in an area of climate change, some work on freshwater, some work on embryos, and some work in the pelagic environment. And I started off by saying that we are in this era of climate change. All of these different climate impactors we know directly affect the heart, and through the heart, they can also affect lifetime fitness. And so I want to end by thanking you for listening, and I'm happy to take any questions.
Lovely. Thanks very much, Holly. Sorry we kept you talking for so long.
No, gosh, I'm sorry. I just kept talking for so long. How long did I talk for then?
Well, almost 50 minutes. So.
Sorry. I can't help it. I thought it'd go faster, but when you try and explain things, they just go slower.
I know.
Well, I apologize, everyone.
No. It's absolutely fine. I'm sure everybody enjoyed it. So we're just going to give a 5. Hey, hello, everyone. Thanks for staying with us and for submitting questions as well. We already have some interesting questions. So I can also see that there are new people who joined to the session. So if you missed the first part or part of the talk that Holly was giving, please be aware that we will be archiving this talk on our YouTube channel. So please come back and subscribe if you haven't already done so to actually watch the whole session. So Holly, thank you again for this interesting talk. I really learned a lot. So I would like to start with audience questions, which I'm going to follow from my phone. So the first question is, are the fish likely to be able to adapt to the new conditions by natural selection?
That's a great question. And some fish we believe will, and some fish we won't. That's we call that kind of the winners and the losers in the games of climate change. The animals that have short duration, short lives and rapid regeneration times are better able to adapt to climatic conditions than animals that have long lives and long generation times, because of course they're able to adapt more rapidly. And so that means that a lot of some tropical fishes and potentially we already are seeing instances of it in some salmonids. The big concern in this area is the elasmobranchs, the sharks and the rays, because they are they have the loser phenotype. They're slow growing. They don't mature until late, and they are overall fairly long lived. And so the concern for those species for adaptation by natural selection is greater than it is for some other fishes.
Okay. So I would like to follow that up as well. So obviously we know that the environmental stresses or constraints the animal has to go through, or us as humans have to go through, you acquire some genetic changes along the line, which you sort of tapped into. So has anyone actually screened the damage that occurs to the genetic material, or the genetic changes? Are there any genetic changes that are discovered within our lifetime to actually show that they've adapted to these conditions?
So I think that we do see speciation and adaptation, and that has been shown really well by colleagues in America on tadpole sculpins adapting to very local conditions as you go up the coast of either of the coasts. So, and that's been shown at all levels and at levels of the gene. So we definitely know it can occur. I think you can kind of think of another way around is thinking of the damage to the genes that we know climate change is having. So we do know that the high temperature and the low oxygen can cause oxidative stress, and that can actually cause instability of the genes and actually incur genetic damage. And whether that genetic damage is carried on to the next generation is something that people are actively looking at right now.
Okay, all right, great. So our next question is there evidence that human impact on the ocean environment is directly affecting commercial fishing? The audience is saying that I guess it would be hard to separate this from effects of overfishing.
Yes, those are difficult things to separate, and I think it's important to remember that combined stressors are often synergistic more than additive. So having both fishing and climate change is together worse than having the additive chance of both of them. I think, but it is hard to separate in some instances, but of course we don't commercially fish all species. And so for species that are both commercially fished and being exposed to changes or difficult climatic challenges, those might be sometimes hard to separate, whether or not the reduction in fish size is because we've been fishing all the big ones or because they've been growing at a warmer temperature and therefore they're smaller catch. Those are harder things to tease out in animals where we have both of those happening. But certainly there are a large enough number of fish species still that are not overly, that aren't fished, only because we don't want to eat them, I guess, at present. But we can then, in those species, we can definitely separate out the climate change from.
Okay, lovely, thank you. So the next question, so two people have just provided feedback on the talk as well. They just said that it was an interesting and great talk.
I think it's too long, I'm sorry, but
no, it was really interesting, and it's really nice to see someone passionate about what they're doing. So are your physiological experiments translatable to conditions in other vertebrates or even humans?
That's a great question. So I think the short answer is yes, most of the time. Certainly, humans or mammals experience the world differently from fish. One, we live in an aerial terrestrial environment, so temperature changes are not as felt as strongly to us as it is in the aquatic environment. And of course, we maintain our metabolism through internal heat, where ectotherms do not. Their body temperature is moves with the temperature of the environment. So in that way, sometimes fish are more, or fish, amphibians, reptiles, they're more susceptible to these climatic stressors because they're not as good at maintaining an internal environment. But certainly, I think, at least in that last example, with the example of the toxicants, the mechanisms that are causing dysfunction to fish hearts are the exact, with aquatic pollution, are the exact same pathways that are being activated by air pollution in human hearts. And so because this was meant to be about environmental effects, and I didn't talk about the air pollution work we do in our lab, but we do look at the similar pathways, and we know from looking at large animal models or human iPSC, so human stem cells that are stem cell derived human cardiomyocytes, we know that the same phenanthrene, the same components that cause the reduction in force and the disruption of electrical activity in the fish does the same thing in our hearts. And we think that they're directly analogous.
Okay, interesting. So just to follow that up in a little bit different direction. So you're measuring the physiology. Is anyone actually looking the behavior of, the changes in the behavior of the habits of the fish? And I'm actually wondering, increasingly we are hearing more attacks, let's say from sharks on people. And as you have mentioned, that they are the losers in this game because they're not going to adapt. So I'm assuming they will be the most stressed out ones. So obviously there is a bias on the fact that hundreds of years ago, if there was something like this in Australia, it was less likely that we heard about it unless you were really living in that region. So do you think with these stresses affecting the physiology of the animals, we will see more changes in their behavior and maybe get more attacks on people, or can they become more docile because of this?
I think that's a tough question to answer. I think loss of habitat and maybe might drive sharks closer to land. But they're usually shy animals, and coming close to humans is usually not what they would want to be doing. So it's hard to definitively link shark attacks with climate change in the waters. But we do know that temperature, hypoxia, and I didn't talk about ocean acidification today, but we know that those do impact certain fish behaviors. There's been studies looking at reef fish and how these different climatic factors impact homing behaviors and predator-prey relationships in reef fish. So I think these are behavioral impacts. We started to look at some behavioral work in our studies, but we tried to do it with our baby sharks, but they were so lazy they wouldn't swim. They just sat there. So